as we delve deeper into the transformative power of genomic medicine, it becomes very clear that its ultimate value lies in translating discovery into tangible human impact. So the next speaker embodies this principle, and our great pleasure to welcome Professor Kym Boycott. Professor Boycott is a leading clinical geneticist and a professor of pediatrics at the University of Ottawa. She holds the prestigious Tier 1 Canada Research Chair in Rare Disease Precision Health and serves as the chair of the Department of Genetics at the Children's Hospital of Eastern Ontario. Her research program is a powerful paradigm for the field which bridges clinical genomics to basic research with a focus on understanding the molecular pathogenesis of rare diseases to improve patient care as well as family well-being. So through her leadership of National Care for Rare Canada Consortium, she is at the forefront of integrating multi-omic technologies to solve medicine's most difficult puzzles. And Professor Boycott will share with us today on unraveling molecular pathogenesis to enhance patient care and family well-being. So without further ado, let's welcome Professor Boycott.
Thank you, everyone, and thank you very much for this opportunity to speak today. It is truly a pleasure to be here from Canada in my role as co-chair of the Lancet Commission, and I really want to thank the organizers for the beautiful opening ceremony. It was extremely touching. So today I'm going to speak to you about diagnosing the most difficult to solve individuals living with rare disease and how we've approached that in Canada. We estimate, when I'm speaking today, I'm speaking about rare genetic disease. It's very important to put that qualifier on what we're speaking about. This is not rare autoimmune disease or rare poisons or something else. This is rare genetic disease. And so we estimate that about 1 in 50 Canadians are living with a rare genetic disease. As we've heard, they're often on a diagnostic odyssey and they're often unidentified and unrecognized. We do have what you would call universal healthcare coverage in Canada, though it is actually a little bit challenging. It is provincially and territorial rolled out by those bodies, but everybody does have access and it is fairly uniform across the country. So my comments are made in that context. All right, I would like you all now to take a few minutes to look at this graphic piece of art that was drawn by an artist in Vancouver by the name of Karina Keeling, actually at a CORD meeting. You might remember Derhain. And I've kept this with me for a long time because what this artist has actually done is really understand the very complex multi-dimensional types of challenges for patients living with rare genetic disease. And she developed this at a meeting that we were sort of presenting many of the things that we're going to be talking about today and the next 2 days of the Lancet Commission around precision diagnostics, precision therapies, and most importantly, global collaboration. And if you look at some of the pieces, you can definitely appreciate the complexity of this world. So what I would like to focus on today is precision diagnostics, and really the top box, which is the understanding the molecular pathogenesis, because you certainly cannot have a diagnosis if we do not understand the cause of your rare genetic disease. I will not be talking about access today, but that is equally as important and complex. You can look at the iceberg on the right, and you can see that we've been spending a lot of time above the water in the coding world, but it is now time that we are starting to go deep under the water to look at the most difficult to identify types of changes that can cause rare genetic disease. So in the middle here, we have this pink girl, and she's the diagnosis. And we are now, through our Care for Rare Canada Consortium, looking at all the different areas. So you can see the girl in purple behind the tree. This is what I see as representative of genes not yet discovered. So she's there, she's visible. We just need to put enough work into it to find out where it is. The boy crying in the box, this is how I feel sometimes. This is where you've been working on a family for 20 years and you cannot solve them, and I'll show you examples today of what I'm talking about. This is where the genetic etiology was undetectable to the technology in some way, despite every effort that one has made. And what I'm actually really finding very interesting these days is the guy hanging out under the earth, I think, and that is where the etiology is more complex or not genetic or a combination of gene and environment. And as we move through the next decades, I think this will be a very important and incredibly interesting area that we're going to delve into, and I'll give you one example of that at the end as well. So everything I'm going to speak to you about in the next 25 minutes or so, I want to acknowledge up front that this is work of the Canadian Collaborative Care for Rare Consortium. We have been together since 2010 actually, so 15 years. We've had multiple large-scale grants across that time where we've distributed all of this funding across the country to 21 sites. We have almost 40 million people in Canada, just to give perspective, and we have about 400 geneticists, 600 or so genetic counselors, and in those 21 sites are the major hospitals in each of the different provinces. The top of Canada there, the territories, all of those rare disease patients are looked after in the province below them. So the first thing I want to talk about then is how we've taken all of our interesting patients and unsolved, undiagnosed patients from across the country. And from the very beginning, with work from Bartha Knoppers, who's also a member of our commission, she built a consent that we used in 2010 that remains active to this day 15 years later and allows that data sharing for discovery which has essentially been unchanged in that whole time, which means she really was at the leading edge of being able to show us how we can share data safely for families for health research. Our data infrastructure is called Genomics 4RD, and it allows us to share in open, controlled, and restricted fashion. But what I want to highlight on the end there is that what do we do with all of this data in there? Because we can all generate lots and lots and lots of genomes, but somebody has to be able to analyze all of those genomes, and we of course hope that machine learning and AI are going to help us with those things, but we have to have a way to dig into that data all of the time. And this is called the Matchmaker Exchange, and it's something that we put together in 2015. And at that time, when we were sitting around the table in Boston, we thought this would last 1 year. We would only need to know this for 1 year, and we would solve all the diseases that there are, all 10,000, as was previously mentioned. But now, 10 years later, it is still going strong, and we now have 170,000 patients that are unsolved connected around the world from 114 countries. And there have been more than 1,000 discoveries attributed to sharing this data in essentially what is a Go Fish, which is a card game, Go Fish experiment. Do you have any interesting variants in patients in gene X? And has somebody else asked the exact same thing? And that is how you matchmake. Now, there are challenges with this, and we will develop, I see over the next 10 years, even more different ways to do matchmaking internationally, keeping everyone's data safe, but still being able to make discoveries for families. And one of our studies really highlighted this in the sense that one can never really share data very well if you don't include clinical data. You just can't. And so one must have information about phenotype, zygosity, segregation in the family sometimes. And some of the challenge we've had with Matchmaker Exchange is because Gene Matcher is the most dominant matchmaker in this platform. And it does not require you to put in any clinical phenotype. So as a result, any gene you put in, you will get a match. 100%, pretty much. And so when you look at our study in 2025, this is our most recent data, we put in 370 candidate disease genes, so genes never associated with human disease before, and we got over 3,500 matches. That's a lot of emails. 60 genes went on, so about 20%, which half are now published. So there's a lot of false positives, let's say. And so it really means that the more information that we can share in a safe way across the country, the more efficient this is going to be. So over the 10 and 15 years we've been doing this. I just want to highlight some of the important work that has happened. And these are all of the genes that we've discovered as part of Care for Rare. And they are actually Tyler Hartley, who's with us and is part of the academic secretariat for the commission, plotted all of these on a cell for us, indicating where the genes are, which means that the families that we're studying with undiagnosed rare disease are giving us insight into an amazing amount of biology. You can see all the different aspects of the cell that are involved there. And what it also shows us is that there's a lot of discovery to be made, even in a small country like Canada, where we don't have huge genomic projects that are running at the level of here, or the wonderful vast set of resources that were just previously described in China. We're relatively small fries, actually, in this, but by being collaborative and sharing, it really has allowed us to make lots of discoveries. So what I would like to go to now is looking at this just showing a couple of highlights around these challenging etiologies and how we've addressed them. And let's start with the guy who's crying in the box again, and we're going to look at genomic rearrangements. And so this is Abby. She has been my patient since she was 2 years old. She's now 14. And I've been trying to solve her since that time. She has had, as you can see on the bottom left there, every single thing one could possibly do in terms of technology. And you can see at 2 years old, the back part of her cerebellum, and then at 10 years old, you can see she has a progressive cerebellar ataxia. And so she initially could stand and sort of walk, and then lost that skill, and is now no longer able to sit independently. And it was PacBio long-read genome sequencing and optical genome mapping that eventually solved her. And so this comes to the point of why the guy is crying in the box. And that is that if you look at the things that long-read genome sequencing that can do, that short-read can't do particularly well, is this one of the highlights there is the structural variance. And so if you look at that, you can recognize that with the short read, base, the 150 base pair of fragments across the top, that you're never going to pick up something as complex as what Abby has ultimately had. Now, Abby has a degenerative condition. She's lost her eyesight. She's lost her sensory nerves. She's now in a wheelchair. She has intractable seizures that cannot be controlled. I would not have thought a structural rearrangement would have caused that. As a clinical geneticist, that's not sort of how I was trained or how I was thinking. But what we did with Abby was long-read genome sequencing and optical genome mapping. We need both of those technologies to solve her. And this is the complexity of what you're seeing here. So when you look at the bottom, this is the very complex rearrangement that we're seeing on the end of chromosome 16. And when you look up into the middle section there, the breakpoints, you can see the multiple back and forth that this has happened. And this is likely the result of a chromothripsis event where the end of her chromosome has been chopped up and it got repaired. And it got repaired in this very unusual fashion, such that she has 3 to 5 copies of many of those genes in that region. And interestingly, we presented this at a poster. As a poster, my postdoc presented it in Milan at the last European meeting, and somebody came up to us and said, we have 11 patients with one of those genes in your region that have 3 copies. Abby has 5 copies of that gene, but they actually have a completely overlapping phenotype. For the 11 patients with triplications, their onset is in their teens. For Abby, her onset was 1 year of age because she has 5 copies and they have 3 copies. And so you can really start to solve something like that, which has taken us 12 years. To begin to understand. And one of the other remarkable things about this is that through our rare disease models and mechanism network for Abby, we were able to build a model of her disease and understand how her cerebellar neurons are dying because of this 5 copies of that gene. I now want to go back to the boy again and look at expansions. And so this is a project that I put in. I started as an assistant professor in 2005. 2007, I wrote this grant. We just solved it. That's how long it took. And so I put in this little, it was for 70,000 Canadian dollars. I was just new to the place, I didn't even have my own lab then. And I wrote this thing because I had seen this ataxia in my clinic from patients of Franco-Ontarian descent, which means French-Canadian families that have been living in the province of Ontario for a very long time. And we tried to say we have lots of families, lots of affected, we were going to go try and solve it. You could even do it back then by traditional methods like linkage, but we could never, ever solve it. And I just have just a video of one of my patients here just showing her ataxic slow walk. So this is a late onset ataxia. Often it's episodic at the beginning and then is there all the time. You can see it has a mild cerebellar atrophy that is progressive with time. It has some unusual downbeating nystagmus, which is quite a feature. And it is slowly progressive, does not limit your lifespan, but certainly does impact your quality of life significantly. And so this is where, again, the long reads come in, because when repeat expansions are super, super big, it is actually quite difficult to see them on short read reliably, though it is technically possible. And so this is the outcome of this. And so we went back and recontacted all patients that had participated in that study after 16 years. We found every single one. And some of them had been followed occasionally, but a lot of them we were able to reach back. And you can see some of our families. And what it's showing there in the long blue is essentially that that's the long repeat expansion. So this is a repeat expansion that's quite huge, and it's going through the gene FGF14. And in my clinic, it accounts for virtually 95% of every French Canadian that comes who has ataxia. And so we probably the most patients in the world, maybe close to Montreal, but it is a ubiquitous ataxia worldwide, and we have over 85 patients now who've been diagnosed in the last 2 years with this condition. Now, why this is interesting is that there's been a bit of anecdotal literature where patients were tried on a treatment called 4-aminopyridine, which I'll call 4AP for short. That is a multiple sclerosis drug that has been used in that way for a very long time. It's quite safe. But things like, 9 out of 10 patients who had a positive response and, their eyes stopped moving and they stopped being so dizzy and they walked a little bit better. And so what's interesting in terms of now taking precision diagnostics to precision therapies is we have a drug that we can potentially now repurpose and do a clinical trial in our 80 patients. This is actually going to be a global trial. And there's also some evidence from mice treated with 4AP who have ataxia that maybe there's actually a slowing down of the degenerative process over time, and it may be protective. And so our clinical trial is now planned for 2026, and will be the biggest recruiting site worldwide, I think. So just moving on to a couple of these other mechanisms, I want to talk about mosaicism for a second. So that's at the top, where things are actually coding, and so in the coding genome, but you don't necessarily see it. And I just want to highlight this as a place not to forget when they're unsolved, especially for dominant disorders like tuberous sclerosis. So this is a clinically recognizable condition. It has clinical criteria, and we have seen over the years many patients who meet the clinical criteria for tuberous sclerosis, but don't have a mutation in either TSC1 or TSC2. And so somebody once said to me, well, maybe there's a TSC3. Well, there's not. These are mosaic mutations in TSC1 or TSC2. And this is just an example of deep sequencing. We're reading now here to 1000x. And that we have to not forget about this as a diagnostic approach for families where we see that they have a typical presentation or maybe even a milder presentation to look for mosaicism. And this is actually very challenging from a diagnostics perspective because it's all about which tissue you're looking at and the chance that that mosaic variant will actually be there. Okay, now I'm getting into a very interesting thing that has come across my research program in the last year or so, which is hypomorphic alleles. So when we are looking at rare disease data, rare disease genetic data, patient data, we are usually filtering to rare variants of 1% or less. Probably what you guys do here as well. What happens if you have hypomorphic alleles? And so I just want to show a picture. This is going to be a little video of Ernie. He was a patient that I saw as a resident in 2003, actually, in Calgary. And we were the first to recognize this particular condition that he has, which we called CAS syndrome at the time, because he had cerebellar atrophy and short stature. So you can just see his significant dysmetria that he has there as he reaches for his little toy. And Mom had always told us that Ernie was pretty normal for the first 6 months of life. And then he sort of had this protracted regressive course such that he then now is in a wheelchair and is unable to sit by himself. So we identified that gene several years later when we had access to exome sequencing. It's being a recessive condition in the gene SLC39A8. That is a manganese transporter, as it turns out. And so now what we know is this is an ultra-rare disease, but we know that individuals who are affected with this have developmental delay and intellectual disability, low tone ataxia. Many of them can have a significant movement disorder and epilepsy. And some of them have short stature like Ernie, who have been very severely affected, and some eye findings. And you can see on the right there, you can obviously see my clinical focus is ataxia. You can see the cerebellar atrophy there, which is quite striking. And what was very interesting to us at this time is then, well, what if we treat these patients with manganese? And at first we were a little afraid to do this because if you actually have too much manganese, it's very bad for you. But that only happens, as we learned, if you're working in a mine and you've been terribly exposed to manganese. But we now know there are 30 patients in the world with this condition, and we have 15 of them on manganese supplementation. And a distant cousin to Ernie, who I just showed you, his older sibling was born with this condition, was diagnosed at 4 years of age and is as severe as Ernie, but their second child also is affected, diagnosed at birth, treated at birth, and is completely normal at 5 years of age. So that means replacing manganese, which let me tell you is dirt cheap. You can buy it for $20 on Amazon, is a very effective way to treat these children. And so you can just sort of look, I don't want to go into all of the things that manganese does, but it is very important as a cofactor for many enzymes in our body, including the beta-1,4 galactosyltransferase. And as a result, your N-glycan profile is abnormal, which we use as a biomarker for this particular condition. Also of interest, it's also a cofactor for manganese, so for SOD dismutase, which is probably responsible for the ataxia piece. So off we went on our merry way with our very ultra-rare disease, sharing our protocols informally across the world, trying to help people find how to source manganese, etc., and get their pharmacy to actually provide it to their patients. What things should we study? How should we follow them? It's really been done haphazardly. There's been no formal trial per se. It's all N of 1 off-label. So then we came along in 2025 and we said, why do we keep seeing a single change in this gene but no second change? And they present just like our other kids. And so we recognized at that time then a hypomorphic allele that's present at SNP. It's present at 5% of the population. And there's functional evidence in the literature that it impacts function. And so when it is in trans with a more deleterious allele, it actually causes the same condition, but in a much milder phenotype. And here's my patient Grace, which I'm going to show you a video of her in a second. There she is at 2 years of age, and you can see her cerebellum. And then at 19 years of age at the back, you can see how much atrophy has happened, even in the mild form of this condition when it is untreated. I have followed Grace since she was 5 years of age, and we did not pick this up until last year, which is a really sad situation where we did not have time to do this. But we've now recognized that this is a very common cause of cerebellar ataxia in children, because we went back to our relatively modestly small Care for Rare consortium and found 12 patients with this. 12. We don't ever find 12 patients with anything in Canada if it's an ultra-rare disease. And so this has been very important. Now here's a picture of Grace. This is where she is now. She's now on treatment. And you can see she walks with an unsteady, broad-based gait, but she's now been on treatment for 2 months or so. She's still titrating up to get her manganese to the normal level, and we really hope that we're going to have some positive improvement even at this age, that we can at the very least, prevent further deterioration. But again, a very cheap therapy for an ultra-rare disease if it's diagnosed. And the last thing now that I want to talk to you about is gene-environment interactions, because I think that this complex inheritance in biology is where these genome projects are eventually going to take us. And we're going to need to have lots of clinical information as well as exposure and environmental information when we think about these things. And so I just want to bring your attention to this group of conditions we call recurrent constellations of embryonic malformations. That is a chunk. So we call it RCEMs for short. And you can see in Table 2 on the right that there are a number of these recognized complex congenital anomalies that we see. And one of the most common ones that many of you have probably heard of is called VACTERL. And VACTERL has major features that you can see there at the bottom, such as vertebral anomalies, tracheoesophageal fistula, cardiac defects, radial ray defects, limb defects, etc. Renal anomalies as well. And so these have been recognized and are diagnosed. You can diagnose them clinically, but they're not inherited. You don't pass them on. And so just to say, what we've done here is epigenetics. And so looking at methylation arrays, you can see that in our 2 cohorts now, we have a discovery cohort and a validation cohort. If you look at the red, that's VACTERL, and the RCEMs on the right-hand side are also VACTERL, and you can see that they're clustering. And so we now have an epigenetic signature that is recognizable and diagnosable for VACTERL, and one of the other conditions with overlap, so they call OAV. So fascinating, but why does this happen? And so you have to start to think back to what the exposures were in pregnancy, because we also know that things like alcohol and valproic acid exposures in pregnancies will also give us an epigenetic signature. So what's happening there that would then move that development in that direction? So all of that to say, this is the challenge that we're in. We're looking at a precision diagnosis for all as our goal. And this is just some, estimates on the right-hand side about where these things might be. It's going to be probably very different than that. But we will need to do a lot of work on all of those aspects in that, on the right-hand side there, because all of them are quite challenging to do at this time. Oops, sorry. And I just wanted to say one more thing, and what we need to do to focus on this then is to bring multi-omics to the diagnostic labs, and I think that's key, right? It's not all about genomes. We actually have to bring some of the multi-omics as well to expand our diagnostic toolbox, and eventually everybody just gets it as first tier, but we're a little ways away from that for now. Not as second tier, which is what we do now and only in research. And I just wanted to say again that the gene-environment interactions, I just am highlighting here this exposome, which I think in pregnancy is going to be a really important thing that we need to capture for these diseases. And you can see here that we're looking at we've identified the methylation signature for valproic acid as well as fetal alcohol syndrome from our same research group. And I just want to add then 2 more things. National data infrastructure, which you've described, and we've heard about earlier today, is important. We all need to do this. We all need to have access, et cetera, for researchers. I don't think I need to belabor that. And the other thing that that our previous speaker also highlighted beautifully was that we need to connect patients to their data and have them contactable, right? So they have opportunities, they understand what's going on with their things. And this is just something we're trying to set up in the States, in Canada, which is a Connect registry, which is basically a national registry for rare disease, so we can find everybody where they are. So just to finish then, I just want to acknowledge all of my collaborators and our Care for Canada Consortium and all of our funders that have funded all of the work that we've presented. Thank you.
Thank you, Professor Boycott. Do you have any questions for Professor? Let's see if we have questions. One question. That will be the only question for this session. We'll discuss further more at the panel discussion. Please, thank you.
Thank you. Brian Chung from Hong Kong Genome Institute. Professor Boycott, I just want to say that your work has inspired a lot of work in the next generations of clinical geneticists. 2 comments on the gene matcher. I'd just like to thank you because of that platform. Our team has actually been involved in the discovery of a few genomic syndromes, and it's really through the work of the gene matcher that you have mentioned, including MN1 and DDX39B that is related to neural disorders. The second thing I want to thank you is your pointing out of the technically challenging variants in the evaluations of patients In the Hong Kong Genome Project, we have evaluated that actually up to 20% of our patients reaching a final diagnosis actually have one of those technically challenging variants. And it's only through genome sequencing, short and long read, that we were able to make that diagnosis. Now, we want to pick your brain on the next important things that probably you're already thinking of how to do. We all know that genomics is very helpful in drug development. And as a matter of fact, those clinical trials that involve genomics actually have doubled the success rate. So how can these databases, multi-omics platforms that you are describing to us help in that process of drug development, not only for patients with rare diseases, but common diseases? What would be your insights?
Yeah, so thank you, Brian. That's a great question. I think I would say, for the short version of that answer, we can talk about it longer tomorrow, that I think that the databases that we share with our partners, including researchers and industry partners, et cetera, they need to be as deep as they can be. So eventually, I think all of our data needs to actually connect into our electronic medical records that have that level of data that researchers and pharma in particular will actually need to be able to look at things like modifiers and the complexity of those types of things that may identify drug targets, that sort of thing. So it's all about the data. Having a list of 5 HPO terms with a genome is not going to cut it, would be my bottom line.
Nowadays, the most topical issue on earth is about AIs, right? So what about the Canadian experience? What about the applications of this kind of smart technologies on helping, to compress or reduce the diagnostic odyssey?
I think there's a number of places that AI can obviously work in the rare disease pathway. Recognition is one of them, and I presented some of that yesterday in an algorithm that we developed called Think Rare that allows the electronic health record to be surveyed each evening, and potential patients that may have a rare, undiagnosed rare genetic disease identified and flagged. And again, I think just like that, it needs to be open source, it needs to be free. I would hate to see somebody patent and try to sell something that is trying to help rare disease families be identified. That would be awful. So I think that's a big piece of this. And the other part about the AI is that the data that we're going to have to share is going to be extensive to have the machine learning and AI actually be able to work its magic. I think I don't know how much the audience knows about synthetic data, but we do have a researcher at my institution who works on synthetic data, which does protect patient privacy, et cetera, but it's very hard to create synthetic data for rare diseases. You don't all have hypertension. And so I don't think that's necessarily going to be our answer. So that means we do need trusted environments to share the amount of data that we would need.
Right. May I delve a little bit deeper that any criteria for joining that network? And in terms of, we have seen the successful national ecosystems, the Peking Union Hospital. You have built in the ecosystem with a collaborative network, engaging several hospitals all around the nation. But in navigating the practical challenges, including data privacy and, or even the national sovereignty, the biosecurity, all these issues, how can you, healthcare leaders or researchers try to, to enhance the kind of a global data sharing? What do you think from the, mainland China perspective and from the overseas perspective that, what are the challenges and how we can overcome to have the sort of the national system to link to the global data platform? What do you think?
The Matchmaker Exchange is the most simple, basic sharing of data one could ever do. And so it's been wonderfully successful over 10 years, but really is not going to be the answer for everything, that's for sure. And so as you increase the level of data sharing from each part, you really need to think about the data from two places. One is the data that's consented for research, and one is the data that is clinical, and how those are shared are different. And I think we actually have a remarkable opportunity with all the rare disease families that we work with to have their data consented for research, and then that allows us to do much more with with it and share it in a much more broad fashion, which is going to benefit everybody. And so I think that is a big piece. And as a clinician, and my colleagues that are clinicians, they'll all sort of pause and say, oh, it's a lot of work to consent a family, you know, we don't have time for this. But I do think we need to make time for that so we can share it in the most generous way. Otherwise, we're stuck with data which is siloed on servers in the country, if not on-prem, and that's not really the way to go.
All right, perhaps I can leave some time to the floor to have interaction with our panelists. Any questions? Yes, the gentleman over there. Can you, mention your name and where are you from?
Rare Care Centre in Western Australia. One of the areas that I'm seeing the greatest advances in data sharing in our own country in Australia is with privacy-preserving record linkage. And we're particularly seeing that evolve with the Indigenous communities who want privacy-preserving ways to share their data, as well as community-level control on how that data is shared. And so there's now a national initiative across all the Aboriginal healthcare providers working together with the federal government on a new data ecosystem, which is underlied by privacy-preserving record linkage and builds in community control on the access to that data. And so just highlight that mechanism. PPRL, privacy-preserving record linkage, is a way to share very detailed information without sharing the identity of the individual. But then can lead to pathways, if potentially you wanted to, you could re-identify that individual for a specific indication. And so I would just highlight that as one evolving mechanism we're seeing developing in the non-rare disease space that could be really powerful for the rare disease space.
Yes, any comments to that?
I think many of us in regions of the world are struggling with Indigenous data and data sovereignty. And I think what that really requires, which I think has been beautifully demonstrated by Australia, is federal government buy-in to set up the infrastructure that's required to do what you've just described. And if you don't have that, it's actually quite challenging, which is what we're facing right now in Canada.
We always talk about the future. So say, for example, beyond better algorithms, more data, what would be the next frontiers in rare diseases? And perhaps what are your biggest hopes for the next phase of this journey?
One of our biggest challenges is actually the data infrastructure and the complexity of the data infrastructure, because as I said during my talk, it's more than genomes and a few HPO terms. It has to be linked, it has to be deep, it has to be patient reported, and it has to all connect. And until we have that, I think many of these conditions that we're trying to improve are going to remain challenging.
Great. So to conclude, we want to develop a patient-centered ecosystem because the end goal isn't just a diagnosis, but a full circle of accessible and affordable care for the patients. And it's about creating a system, just listening to the two presentations from different models, from genetic testing to affordable drugs to ongoing support for the patients that truly ensures no patients leaving behind. So let's get another round of applause for our amazing, fantastic speakers.